In 2020, a team of oceanographers from the Woods Hole Oceanographic Institution in Massachusetts published in the scientific journal Nature the results of a study that transformed our understanding of the limits of life on the ocean floor. Using a specially designed sediment drilling system mounted on the ROV Jason, the team extracted sediment cores from the floor of the North Pacific in a low-productivity region known as the North Pacific Gyre at a depth of between 5,500 and 6,000 meters. The sediment cores, which reached up to 6 meters in length and represented between 80 and 100 million years of sedimentary deposition, contained in their deepest layers corresponding to sediments deposited between 80 and 100 million years ago during the Cretaceous period, something scientists did not expect to find: living, metabolically active bacteria that had remained buried under tons of marine sediment for tens of millions of years, consuming the dissolved oxygen that slowly penetrated through the sediment's pores from the bottom water. It was the discovery of a completely unknown deep-sea biosphere, buried beneath the ocean sediments, which had remained invisible to science for all the time humans had been studying the ocean. Subscribe and share. Let's begin. The North Pacific Gyre is one of the least biologically productive areas in all the world's oceans. It's what oceanographers call an ocean desert. The circular currents of the subtropical gyre create a zone of stagnant water where nutrients are depleted in the sunlit surface layer and not replenished because there are no active vertical mixing mechanisms to bring nutrients from the depths. The result is a zone of crystal-clear, deep and beautiful blue water that produces very little phytoplankton and supports an extraordinarily sparse food chain compared to the productive waters of coastal seas or upwelling zones. So little organic material reaches the ocean floor in this area that the rate of sediment deposition is only 1 or 2 millimeters per 1,000 years, which explains why 6 meters of sediment represent tens of millions of years of history. Scientists had assumed that the deepest layers of these sediments, so ancient and so depleted of organic material, would be virtually biologically sterile. The surprise began during the processing of sediment cores aboard the research vessel. Standard protocols for analyzing ocean sediments include bacterial viability tests that detect the presence of living cells in the sediment. In the surface layers of the cores corresponding to recent sediments, the presence of live bacteria was expected and confirmed. As the analyses progressed to deeper, older layers, the researchers expected to find a gradual decrease and eventual disappearance of signs of life. Instead, they found viable bacteria in all layers of the core, including the deepest ones corresponding to the late Cretaceous. And when these bacteria were cultured under laboratory conditions, they not only survived but began to multiply, going from the tens of cells per cubic centimeter typical of deep sediment to billions of cells in just a few weeks. The most immediate implication of this result was that these bacteria were not simply dormant or in a resistant spore state, as might be explained if they had merely survived in a state of latency for millions of years. They were metabolically active, consuming oxygen and producing carbon dioxide, albeit at incredibly slow rates. The team calculated that the metabolic rate of these bacteria was between 1,000 and 10,000 times lower than that of the same type of bacteria under normal laboratory conditions. This extreme metabolic slowdown was the key to their survival. At these consumption rates, the minimal energy resources available in the sediment were sufficient to sustain basic cellular processes for geological periods of time. The researchers described this mode of life as existing at the absolute limit of what is considered active life, on the threshold between metabolism and complete dormancy. The extent of the discovered ecosystem was difficult to estimate from just a few sediment cores in a specific area of the North Pacific, but the researchers attempted to project the data onto a global scale. If the subsurface ecosystem of bacteria in ocean sediments extended across the entire seafloor in low productivity zones, which represent more than 70% of the total ocean surface, the total biomass of that buried ecosystem could be comparable to that of all life in the surface oceans. It was an extraordinary claim, and the authors themselves presented it with all due caution, but the data from the North Pacific cores and complementary studies in other ocean regions consistently pointed in that direction. Life on the ocean floor was far more abundant and far older than anyone had imagined. The discovery has implications that extend far beyond marine biology. For astrobiology, the scientific discipline that studies the possibility of life on other planets, the existence of organisms capable of surviving and remaining metabolically active for 100 million years under conditions of extreme energy deprivation radically expands the range of conditions under which life could exist on other bodies in the solar system. The ocean moons of the giant planets in the outer solar system, such as Europa around Jupiter or Enceladus around Saturn, could have conditions at the bottom of their subsurface oceans comparable to those of the North Pacific Ocean. Cold, dark, with scarce energy resources, and subject to enormous pressures. If life can persist for 100 million years under these conditions on Earth, it could potentially do the same in these extraterrestrial oceans. The director of NASA's astrobiology program, in an interview given shortly after the study's publication, described the finding as one of the most significant for the search for extraterrestrial life in the last 20 years. Like, subscribe, and share. The scientific community responded to the study with a mixture of enthusiasm and methodological skepticism, as is typical with findings that challenge established categories. The main technical questions centered on the possibility of sample contamination during the drilling and core extraction process. If bacteria from the surface layers of sediment or seawater accidentally contaminated the deeper layers during drilling, this could explain the presence of live bacteria in the oldest sediments without needing to invoke survival for millions of years. The Woods Hole team addressed these objections by describing in detail the contamination control protocols used, which included the analysis of chemical tracers that detect any mixing between layers during the extraction process. The reviewers of the study in Nature concluded that the contamination controls were adequate and that the results were valid. Expeditions following the original 2020 study have expanded the findings to other ocean regions. In 2022, an international team including researchers from Japan, Germany, and the United States published drilling results from the South Pacific confirming the presence of living bacteria in sediments over 100 million years old in that region, with metabolic characteristics similar to those found in the North Pacific. In 2023, analyses of sediment cores from the South Atlantic added further evidence that the subsurface ocean ecosystem was a global phenomenon and not a local peculiarity of the North Pacific. The picture that emerges from all these studies combined is one of a deep ocean biosphere spanning the globe, buried under hundreds of meters of sediment, extremely slow in its metabolic processes, but persistent for periods of time that outlast any other known form of active life. The implications for understanding the oceanic carbon cycle are also significant. Deep-sea bacteria consume oxygen and produce carbon dioxide, and although their metabolic rates are extremely low, the global and temporal scale of this process could contribute to the oceanic carbon cycle in ways that current models do not account for. The ocean absorbs approximately 25% of the carbon dioxide emitted by human activities each year, and the accuracy of this estimate is critical for climate models that predict future global warming. If the deep-sea bacterial ecosystem alters the oceanic carbon cycle in ways not included in current models, this alteration could affect the accuracy of climate predictions in ways that scientists are still evaluating. What the oceanographers at Woods Hole found buried beneath the floor of the North Pacific in 2020 was one of the greatest expansions of biological knowledge in the 21st century. An entire ecosystem composed of organisms that have been active since before the dinosaurs went extinct, hidden under sediments accumulated over 100 million years, sustained by energy resources so scarce that no previous biological model would have predicted they could support active life. The ocean, which covers 70% of the planet's surface, still holds more secrets than science has even begun to imagine. The ROV Jason, the vehicle that made it possible to extract the sediment cores that led to this discovery, is one of the most important oceanographic research platforms currently in operation. Operated by the Woods Hole Oceanographic Institution, Jason has conducted more than 500 dives since its commissioning in 2002 and has been instrumental in dozens of major scientific discoveries in oceanography, marine biology, underwater geology, and archaeology. With a diving capability of up to 6,500 meters, it covers most of the average depth of the world's ocean, which is approximately 3,800 meters, and can operate for consecutive days on the seabed with a suite of scientific instruments that is regularly updated to suit the needs of each mission. The sediment drilling system developed specifically for this campaign, capable of extracting cores up to 6 meters long from sediments of varying consistency without mixing the layers during extraction, was a technical innovation that opened the possibility of this type of high-resolution stratigraphic study on the deep ocean floor. The publication of the study in Nature in 2020 was followed by a debate in the scientific community about the implications of the findings for the very definition of life. The organisms found in the Cretaceous sediments operate at such slow metabolic rates that the distinction between active life and prolonged dormancy becomes philosophically complex. If a bacterium takes 1,000 years to complete a single cell division, is it living or merely surviving? The answer from the biologists on the Woods Hole team was pragmatic. As long as the organism consumes energy, maintains the integrity of its cell membrane, and can respond to changes in its environment, it is alive in any biologically meaningful sense of the term. But the underlying question about the limits of life, about how much an organism can slow down before crossing the threshold into death or indefinite dormancy, remains a subject of debate and active research in several extremophile biology labs in Europe, the United States, and Japan. The connection between this discovery and the debate about the origin of life on Earth is also noteworthy. One hypothesis about the origin of life on our planet, known as the RNA world hypothesis, suggests that the first life forms arose in low-energy, resource-scarce environments, possibly in the sediments of the early ocean more than 3 billion years ago. If the organisms present in the deep sediment represent a biological lineage that has maintained extremely conservative metabolic modes since very early times, their study could provide indirect information about the conditions under which life arose on Earth. Astrobiologists working on habitability models for extraterrestrial oceans see in these organisms a window into the most basic possibilities of life's biochemistry, a demonstration that life can persist and thrive with minimal resources for periods of time that defy conventional biological intuition. Like, subscribe, and share. See you next time.